Hello, my name is Connie Liu. I'm an undergraduate at MIT, and today we're going to be talking about viruses. So first off, let's kind of look at the structure of a virus. So this is what one type of virus looks like, but there are two main parts about a virus that you need to notice. First, there's this outer coat. It's made out of protein, and it's known as the capsid. So this is where all of what's inside here, genetic material, is stored. And this genetic material can be DNA, RNA, really any type of nucleic acids that can store genetic information. So those are the two main parts of a virus. So the way that a virus infects a cell at first is pretty simple. Draw a small virus right here, and then it's on a cell. So this virus is going to essentially take its genetic information and then inject it into the cell. So this genetic information is now inside the cell. So there are two main things that the virus can do. It can enter a lytic cycle, or it can enter a lysogenic cycle. So first, we're going to focus on the lytic cycle. The lytic cycle has to do with the virus completely destroying the host cell immediately, and any cells that only have the lytic cycle are known as virulent viruses. And now let's look a little more into what happens during the lytic cycle. So during the lytic cycle, you have the DNA inside the host cell. This DNA commands all of the mechanisms within the cell for DNA replication, and then it's able to replicate itself multiple times. And that includes the protein parts as well, because it can command the ribosomes. And then using all these parts that it just created, it creates an army of new viruses. And then from there, it can burst the cell, or what's known as lyse the cell. And all of these viruses come out of the host cell that's now dead, and then go infect even more cells in the colony. So that's how the lytic cycle works. Basically, the DNA of the virus takes over the cell, creates a bunch of itself, and then lyses the cell, destroys it, and then all of these new viruses that it created using the host cell parts are released into the outside where it can infect even more cells. Now, if we look at a lysogenic phase, we're going to be seeing that the replication and the virus infection happens a little bit differently. So the same thing happens in that the virus DNA goes into the cell. But what it does here is that it actually incorporates itself into the DNA of the host cell. And then by doing that, every time that this cell replicates, it's going to be able to still have that segment of DNA within those cells. So this cell would continue to replicate more and more. So there would be a ton of them, and they would all have this segment of virus DNA. And then at some point, a trigger will happen. It's really unsure what these triggers usually are, but some type of trigger will make these viruses suddenly become virulent, and then they will enter the lytic cycle. So essentially, the lysogenic cycle is just another way to replicate a bunch of viruses and then do the same thing as before. So then these will, the virus DNA will take over the parts of the cell that are involved in replication, create a bunch more viruses, and then burst through these cells, and then go infect even more cells. And this would happen to all of these cells now. So as you can see, that scales it up quite a bit. So in that case, those are the two ways that that viruses can infect a whole body of cells. And this is really important in that it can cause a lot of really bad diseases, say the flu, or that's short for influenza, is a virus that infects a lot of people during the flu season. And a lot of other viruses are involved in causing disease and things like that. So by being infected by it, it could cause a lot of issues.